Morning and warm welcome 10th standard for your science and technology part B lecture of the online learning teaching session. Last time we have started with lesson number 6, animal classification. In that we started with the introduction, then the history of animal classification, how did this come up, what are the benefits, why do we study this classification, why it is done, then how was traditionally this classification was done on the basis of chordates and non-chordates characteristics. Absence and presence of notochord we studied. Good. Then we studied the different phylums under non-chordates. There are 10 different phylums. Under chordates you have just one phylum, that is chordata. Under that one phylum you have 3 subphylums. One of that, vertebrata, has 6 different classes. I also gave you the abbreviation how you can remember the classes of the subphylum vertebrata. Then we moved on to the criteria for the new system of classification. So first criteria was body organization. How is the body of the different organisms organized? Then body symmetry, and we shall start with germ layers. Now when you study the multicellular organisms, there are these germ layers which are formed during the initial period of your embryonic development. We have studied how embryos are formed from the zygote, how zygote is formed from the fusion of male and female gametes, right? So when this embryonic development is taking place, where there is a group of cells, gradually these group of cells mature into these germ layers, and these germ layers then gradually develop into your tissues, then further development of your organs, and organ system takes place. Now there are 2 types of germ layers. Okay, on the basis of that you have 2 different types of organisms, diploplastic and triploplastic. Diploplastic organisms will be those who have only 2 germ layers, that is endoderm and ectoderm. Endo is the outer one, endo is the inner one, and ecto is the outer one, right? Endo means inside, ecto means outside. Whereas the triploplastic organisms will be having 3 germ layers. You can easily understand by the word di and tri. Di means 2 in biology and tri means 3. So we higher animals have our trypoblast. We have endoderm, the most internal germ layer, mesoderm, the one in the center, and ecto, the one at the outside. Okay, you can see the difference looking at the diagrammatic representation given on your screen. So when you study these animals, some animals will have only 2 germ layers. Okay, your cnidarians, the third phylum of non-chordates. Okay, otherwise in most of the remaining animals you will have these 3 germ layers, that is endoderm, ectoderm, and the center mesoderm. Clear? The 4th criteria is body cavity. Now cavity is also called as coelom. What is the coelom? It is the space between the body and the internal organs. Okay, now again this is formed, this is present from the time of the initial period of the development of embryo. Okay, this body cavity is formed, you can see. Now this is formed from the mesodermal layer. Clear? So those would be your high animals, the triploblast. Yes or no? Now whichever phylum comes after Annelida, that we will be studying in detail when we start with the non-chordates. So all the animals that are present in phylum Annelida and all the phylums or all the organisms that are present in the phylums after the phylum Annelida would be having this body cavity, right? So these animals which have true body cavity developed from the mesodermal layer, they are called as eucoelomates. Okay, eucoelomates. Eu means developed. It comes from the word eukaryotic, if you all remember. Good. Now sometimes body cavity is absent. So this you will get to see in the animals from phyla Porifera, Cnidaria, and Platyhelminthes. Don't get confused with the animals. You'll be studying which animal is present in which phylum. Right now, you just understand body cavity is present in which phylum and it is absent in which phylum. Whereas, in many of the cases, okay, such as Aschelminthes phylum, they will have the body cavity, but it is not formed from either of the ways that is given before, just like eucoelomate or acoelomate, right? So these animals are called as pseudocoelomate. Okay, it is pseudocoelomate. So the ones with the true body cavity, those are eucoelomates. The one who do not show presence of body cavity, those are acoelomate. That is, coelom is absent, cavity is absent. And pseudocoelomate, it is not actually true, but it is not absent as well. Okay, I hope this characteristic is understood by you all. Let us see the next one. Okay, now before we move to the body segmentation characteristic, let's just look at the pictorial presentation. See, the digestive tract, which is formed from the endoderm. I said you that from these you will have your tissues, your organs and organ system being developed. Body is covered by ectoderm. Ecto means the outside layer. Okay, and from mesoderm you will have the tissue layer lining your cavity. You can see the cavity as well. Coming to pseudocoelomate, body is covered from the ectoderm. The muscle layer formed from your mesoderm. Okay, and the false cavity that you can see in the pseudocoelomate. And coming to the acoelomates, I hope it is very clear the cavity is not present. Okay, tissue filled region that is formed from the mesoderm. The last characteristic is your body segmentation. Now, when you choose this feature or this criteria to study the animal, you will just have to superficially look at the animal and you will be able to distinguish. You can see small segments or small units like throughout the body of the animal. Okay, so this what you can say, the body of the animal, when it is divided into your smaller units or smaller segments, okay, that body is called as segmented body. The organism on your screen is very commonly seen, and in this season you will get to see in huge numbers. Earthworm, right? You need not have a microscope or magnifying glass to look at this or to study this organism. You can just superficially look at this earthworm and you can identify the segments present on its body. So you can say earthworm has a segmented body. Then you will also get few animals from phylum Annelida which would be having such segmented body. So the ones those are having segmented body, these are placed in the phylum Annelida. Okay, so on the basis of these 5 different criteria, the animal classification is done, and then you have your total chart ready to study and understand. See, this way the organisms are studied with the help of the criteria that we studied, and then the animals are classified and placed in particular phylum. So all the animals with the cellular grade organization and asymmetrical body, okay, they'll be placed in the phylum Porifera. Then the animals which show radial symmetry and are from the tissue organ or organ grade system, they would be placed in the phylum Cnidaria, right? Then you either, and also there will be some organisms which belong to the tissue organ system grade body organization, but they show bilateral symmetry. Then you have body cavity to study. Okay, so as the animals or the phylums get going higher and higher, you would be able to see more and more criterias being present. The animals would be much more developed. Okay, and then this is how the classification is done from lesser developed to the totally developed animals. So that is why I told you that you will have to remember the order of the phylum. You cannot mess up with that, because the lesser developed or the least developed organisms are placed in the lower phylum, whereas the totally or completely highly developed organisms are placed in the topmost phylum or the topmost class that we'd be studying now. Okay, so I hope till now whatever we have studied is very much clear to you all, and if you remember this criteria, this method of classification, I am sure this is really going to help you in studying the characteristics of the animals, because then it would be easy for you also to differentiate or to place the animals, okay, as a scientist have studied, right? So let's see now next what we have to start with. Now from here we start studying with the phylums, okay, and on the basis of the criteria how the animals are being placed in one particular phylum, that will be studying now. First we start with the phylums of non-chordates. The first phylum is Porifera. Now to remember the name, pori, that comes from pores, okay, so simple, the animals present in this phylum will have pores on their body, small openings on their body, hence the phylum is Porifera. Now their body plan is very simple, that is their body makeup is very simple. These are called as sponges, okay. Now they will be having huge number of pores on their body, hence Porifera phylum. These pores are called as ostia and oscula. Clear? You will have to remember the diagrams in your mind. You can also practice them regularly along with the labeling, that will help you to remember. Now ostia and oscula. These animals of Porifera are aquatic, that means you will find them in water bodies. Most or majorly are marine, you will find them in marine waters, and very few are freshwater dwellers, okay. The body symmetry is asymmetrical, that is there is no imaginary excess kind of anything, and hence you will not be getting two halves in any case. They have special type of cells which are called as collar cells. You see on your screen in the second organism, cycon sponge, okay, that is your cycon sponge. You can see osculum, collar cells, ostia, right? Good. They won't be having any they do have substratum. Substratum is what? In the cycon sponge, you can see at the base they have a support like structure, that is their substratum, that is called as substratum. Now when they have this substratum, they cannot move from one place to another, that is they are not able to locomote, they show no locomotion, and hence they are sedentary, that is you will find them always on one place. They won't be moving from here and there because they are attached to a base, attached to a substratum. Now these are supported by spicules or fibers called as spongin fibers. Now these spicules are made up of calcium carbonate or silica, okay. What is the eating habit? These animals feed on smaller ones than them. They will usually take in the smaller organisms along with the water present around them. I told you these are aquatic organisms. Good. Now how do they take in the food? They'll take in the water and the food. Okay, it is taken through ostia and it is given out through osculum. You can see ostia and presence of osculum on the screen. Good. Now what is the reproduction type? Reproduction can be asexual, it can be sexual. They have the capability of reproducing sexually also and asexually by budding. Okay, and one more feature of these organisms in the phylum Porifera is regeneration. They have the capability to regenerate. This regeneration you will be able to see in lizards. You might have seen sometimes the tail of the lizard is cut off, and after some time they have the tail back again. So that is the regeneration ability of these organisms. Okay, but your lizard does not belong to phylum Porifera. Please do not get confused. Okay, so examples of your Porifera are Euspongia, Hyalonema, Euplectilla, etc. Okay, also on the screen you have two. For better understanding, I would recommend you all to practice the diagram of these organisms and label them well, so it will help you to study. In this case, you can very well remember the osculum, collar cells, ostia, and then you can write in your own words, right? All the criteria that we studied, body organization, then body symmetry, yes, this all are present in this phylum. Okay, so now you get it how the animals are classified based on that criteria. Good. Okay, next let's prove to next phylum. Phylum Cylindrica or Nidaria. Now, to remember Cylindrica, I'll tell you the body of these organisms is cylindrical. Okay, hence Cylindrica. Better to remember? Okay. Now, either you will have the body animals of body shape cylindrical or umbrella shaped. Okay. Now, cylindrical shape animals are called as polyp, and those having umbrella like shape, they are called as medusa. Okay, cylindrical body, those are polyp. Umbrella like body, those are your medusa. You can see three different organisms from the phylum Cylindrica on your screen. Sea anemone, it is cylindrical. Hydra, again cylindrical. Whereas coral, if you observe, it is umbrella shaped. Even you can go and Google jellyfish. Okay. Now, most of the animals from this phylum are marine. You'll be finding them in waters, either in the fresh water, either in the marine water. Okay, but those are aquatic. Symmetry, they are radially symmetrical and diploblastic. From here, presence of germ layers are seen. So these are diploblastic, which means they have only two germ layers, ectoderm and endoderm. There is no mesoderm. Clear? Now, speciality of these animals or something different. They have nidoblast. Okay, look at the diagram of Hydra for better understanding. You can see tentacles, those are present around the mouth, right? Now these tentacles are very useful to capture the prey, because you can see the mouth is surrounded by the tentacles, right? So whatever prey they would want to attack, they can capture the prey from the with the help of tentacles. They'll surround the prey with the tentacles and they will capture it and put it into the mouth. The nidoblast, which would be present on the tentacles. Okay, you cannot see on the diagram here, but there are small structures present on the tentacles. They are termed as nidoblast. These nidoblasts will inject the toxin in the body of prey. Now, toxin is harmful substance. Okay, so that once injected in the body of prey, it would kill the prey, and that would be the food of Hydra or whichever other organism is present in phylum Cylindrica. This is how they can capture their food, they can attack their prey, they can protect themselves from the predator as well, right? So this is the special feature of the organisms in the phylum Cylindrica. Physalia is one of the example. So you can study these examples in more detail, or you can just have a check on how these animals look alike from the Google. Okay, so that will give you a better understanding. Clear? Next phylum, Platinthes. Now the organisms in this phylum are flat. Hence, in general language, they are also called as flat worms. Okay, you can see on your screen, they are slender from the apex and the tail region. They'll be a bit narrow as compared to the center of the body. Okay, and they are flat. Most of them are endoparasites. Endo, inside, parasite, which are dependent on others for their food. Now, endoparasites means you will find them inside the body of an animal, of higher animal. They survive there for their food. Few are free living and aquatic. Some you can find in the water as well. Okay, this tapeworm and few other worms from this phylum, you can find them inside the stomach of higher animals. Okay, they survive there by eating up the food from the stomach of that particular animal. Body cavity is present? No, they are acoelomate. And symmetry is bilateral, that is, you have one imaginary axis and it would divide into two half, right? What about the germ layers? They are triploblastic. They are much developed than the previous one. They'll be having all three germ layers: endoderm, ectoderm and mesoderm. Good? Easy to understand? Yes. These animals are hermaphrodites. What do you understand by hermaphrodite? Both the male and female reproductive organs are present in the same animal body. You will not find separate male planaria, female planaria, male tapeworm, female tapeworm. Both the male and female reproductive system you will find in that one particular animal body only. Such animals or such organisms who have both the reproductive systems in one single body are known as hermaphrodites. Okay, clear? Here the examples are liver fluke given on your screen, planaria, tapeworm. Okay, so this is your phylum Platyhelminthes. Before moving to next phylum, let us look at two surprising information that we have to see. First is bath sponge. Now this bath sponge, you can see on your screen how it looks like. These are black colored and they are somewhat round in shape. Okay, these are usually made of the fibers of a protein, and the name of that protein is spongin. Because of this, they have good water holding capacity. They can hold water and in huge amount. Basically, because of this feature, previously in olden days, these sponges were used for bathing because they can hold good amount of water. And other than that, they were also used for manufacturing pillows and cushions because they were pretty soft. Also, they since they are wetting material, so they were used for sticking postal stamps. And when you count the currency notes, you need your fingers to be wet. So these sponges were used in olden days. Second information is about the coral reef. This coral reefs you might have seen on the Discovery channels when they show you about the oceans, okay, the deep ocean beds. You might have seen these coral reefs because you don't find them obviously near the seashore, right? These are very very bright in color. Yes, so, and one of the important feature about them is they live in colonies. You will not find them single present. Okay, one coral is one corner of the ocean, other coral no. You will find them, they are present in group, they are present in clusters. Okay, so that is called as coral colony. And these corals belong to Cnidaria or Cylindrica phylum that we studied just now. Precious stone, which is called as coral, or in Marathi it is called as pora. You might have seen. Okay, and the coral powder, that is bhasma. Okay, the powder made out of coral, they are used in the Ayurvedic treatment derived from these coral. Okay, if you want, you can collect more and more information about these. You can have a look on more types of bath sponge, more type of corals and other organisms that we have studied till now. Okay, so the more examples you remember from a particular phylum, the more it will help you in studying the features. Okay, fine, let's see. So we will have to wind up for the day because the time is up, and we shall continue with the lesson next time. This lesson is pretty interesting. Just pay attention. Look at the colorful pictures of the organisms. They are indeed very beautiful. It helps you understand the nature, the different kind of animals around us. Yes. So we shall continue next time. Till then, stay safe, stay home, take care, keep studying, keep revising yourself. Goodbye.